During a myocardial infarction, up to 1 billion cardiomyocytes may die, and this will create an irreversible damage in the heart muscle. And these damaged cells, they are replaced by fibrotic scar tissue, and this leads to a stiff heart, reduced compliance, and also reduced contractility, and then eventually a decline in cardiac function. And many of these patients, they also progress into end-stage heart failure. Our ambition is to form new cardiac tissue via remuscularization of the heart, so that we in the future can cure patients with heart failure.
So after a heart attack, it's extremely hard for adults to regenerate their lost cardiomyocyte. So what we're trying to do is, how can we replace this heart tissue when we don't have it innate, endogenously in us? So this mechanism of cell repair is highly coordinated, which involves mainly the cellular process, works in a synergy to generate a new cardiomyocyte without any tissue scarring. So for the HVPs, what we're trying to do is that the HVPs are able to generate new myocyte during the heart's early development. That's what we're trying to mimic, since these are progenitor or precursor cells. We want to use a precursor cell because of the fact that they would integrate a little better within the heart tissue. They would hopefully not cause too much arrhythmia, and that they'll mature together and sync together with the rest of the host tissue. So in 2014, when I joined Kenneth Chan's lab, I took over a protocol from a postdoc in the lab who was working to develop a way to make billions and billions of ventricular myocyte. And from that protocol, I decided to put it into the kidney capsule of a mouse to see what happens in a living animal. What would it do? Because we know that in a dish, we can form muscle patches. So miraculously, in 2 months after we put it into the mouse kidney, we form a big, massive ventricular tissue patch. So from there, we want to know what is the mechanism behind it and what happens if you put it actually into the heart after a heart attack. So we give the mouse heart a heart attack, a mini heart attack, and we put these cells, HVPs, into the heart. And what we found is that there's a functional improvement in the mouse after we put them these cells. So that was the beginning of the first paper. And after that, we decided to see what is the mechanism behind it, because we know that there's a functional improvement, but we don't exactly know what is it that's causing it, the triggering the functional improvement. So from there, we decided to collaborate with Munich to dissect out the actual functional mechanism behind the HVPs.
So we knew a lot about how HVPs are able to really form functional ventricular cardiomyocytes. So we wanted to know, is it possible to also investigate the HVPs ex vivo, to not have to do this in vivo studies and all the animals, but to join forces or also save resources and just try this in an ex vivo setting where you could just closely follow the HVPs, how they would act and react in this situation. So the very first step was really that Kylie sent me vials of HVPs that I thawed and just directly put on my NHP heart slices that I cultured ex vivo.
So what they did was that they have a little mini chambers with tissue slices in which we can put the HVPs on one side, and we have a injury model of the other side, and we see that the HVPs actually migrate, actually senses, the cells itself can sense that there's a tissue damage on one side and it move by itself to the other side to try to repair the damage.
It was just an observation study at the beginning. So we were just looking, are they able to attach? Are they even able to migrate? And it was really fascinating to see that they indeed they evenly distributed, they migrated, and they just perfectly aligned. And with our system, we could see that the contractile performance at the beginning decreased, but then with the HVPs in this setting, it regains again the contractile force. And it was quite cool to see that, not even by eye, to see in the microscope that the twitching, but then also really the contractile force of this whole patch regained. And we have seen, okay, the HVPs really do their job. They are able to restore or to functionally repopulate native cardiac tissue. So we introduced this radiofrequency ablation technology that really made a clear and a stable injury into the tissue. And we see that the HVP is opposite the tissue. You can also see it in the paper on figure 3. It's where we were able to follow the green signal of the cells. So it was really able to follow their migration. And it was quite fascinating to see that we had this street of migration that was just directly directed towards this RFA injury. So we had this migration street following, and then the green signal of the cells really enriched in this RFA injury. So we could really observe the cells seem to directly have a cue and a signal towards the injury, where they also follow that signal and go there. So we were fascinated to see and wanted to know what's causing this signal. So first of all, we sectioned a lot of tissues and looked, okay, what's going on? Are the cells really there where we expect them to be? And we have seen, okay, the cells really enrich in this RFA injury. But there was another finding that was quite intriguing. So we have seen, okay, the beginning, there is also another cell population following this migration street, and it was the cardiac fibroblasts. So we identified that there's this population of activated cardiac fibroblasts that really first go into this injury and then seemed to attract the HVPs. And this was then, we tried to find out which signaling pathway, and it was kind of a guessing, and it was kind of a literature research in a series of signaling blockages until we really identified, okay, this has to be the CXCL4, CXCL12 axis. So it's working that we found out and presume that the cardiac fibroblasts were activated by this injury are the first ones to come there and receive their cues. But then by this, they secrete CXCL12, and the CXCR4 positive HVPs are able to respond to this. They have this receptor that is quite unique, and for instance also differentiated cardiomyocytes, they don't have that anymore. They don't express the CXCR4 receptor, which is why we think that the HVPs are the perfect cell population to respond to this cue, since they have this receptor. So from these results, coming from the observation and knowing, okay, the HVPs can really regenerate cardiac muscle in vitro or in this ex vivo setup, we were curious to see, okay, is this an effect that is only occurring ex vivo in our model system, or is this really going on in vivo, and can we reproduce these results?
We have performed an investigational study in Yucatan mini pigs, and in this study we injected HVP cells directly into the cardiac muscle.
At Procellum, we transferred the process to produce HVP cells in a laboratory, in a dish, in a research and development laboratory, and developed a scalable process to produce large amounts of HVP cells required for the chronic pig study. In addition to that, we developed a suite of analytical assays that were used to monitor and control the quality of the product during the manufacturing process and the quality of the final product, so the HVP cells, in terms of purity and identity and function.
So we delivered 1 billion cells with 15 injection sites, both into the border zone and into the infarct area. And then we had 3 months follow-up. So what you can see there in the histology images, so if we start to the left, is that you have an impressive remuscularization of the scar area and nice formation of cardiac patches within the scar. In the second panel, we have the staining for MLC2V, that is the ventricular cardiomyocyte marker. And we have noted that we have more than 90% of the HVP cells that develop into ventricular cardiomyocytes. In the third panel, it's the N-cadherin staining that is highlighted in blue. So here we see staining in the intercalated disc between the cardiomyocytes. So we have some integration here. And in the last panel to the right, it's staining for the vascular, the endothelial cell marker CD31. And we also know that the majority of these blood vessels, it comes from the pig tissue. And so we have also noted that after 3 months, we haven't detected any teratomas, and that's of course very important also from safety perspective. We also followed the cardiac function over time, so we did MRI measurements at baseline and at 3 months follow-up. And what you can see in this graph is the infarct volume, and you can nicely see there that HVP cells reduce infarct volume from baseline to 3 months, and these data goes nice hand in hand with the histology images we just looked at. And at the same time, we also have a reduction in ejection fraction in the vehicle treated over time. So you can see that the vehicle treated pigs, they decline in cardiac function. The HVP cells, they are attenuating this decline in cardiac function in a very nice way. And looking at the global longitudinal strain graph here, we can also see that the vehicle pigs, they are worsening over time, while the HVP treated pigs, we can see an attenuation in this worsening.
The results show that human embryonic stem cells can be used as a starting material to produce human ventricular progenitor cells with potential clinical application. The process that we developed at Procella to produce HVP cells can be used now as a foundation and as a baseline to investigate further developments to develop an affordable manufacturing process to produce HVP cells at large scale for clinical application.
What we found from this, the conclusion of the paper, is that we found the HVP progenitor's unique ability to heal and repair injury area, in that it's a highly coordinated mechanism between sensing how the cells can sense an injury in one's end, and that what we can do to migrate to actually move towards the injury area to heal it, to repair it, and to repel the fibroblasts.
We have now generated very clear data, both in rodent and also in large animal, that the HVP cells can improve cardiac function after 3 months.
I think we have obtained enough data to know, okay, this cell therapy can be a safe and a reliable source to really help to cure end-stage heart failure.
Cell therapy is a very exciting area, and it brings opportunities of curing diseases and improve the quality of lives for patients with heart failure.